Thank you so much for that kind and generous introduction, Shri. It's a pleasure to be here, and I've had great meetings and stimulating intellectual conversations with people so far, so thanks for the opportunity. So I'm told I should give a bit of background as to why I'm here. And I think I'm here because I have a baby sister who's 9 years younger than I am. And watching her transition from a flailing, helpless newborn to an interactive child was really striking for me as a child. And I was puzzled by how she learned to make sense of words without anybody actually teaching her the meaning of words. And this led to what I would now recognize as some pilot experiments. So I would pick a word that I didn't think she knew, and the word was inteligente in Portuguese. So we grew up in Brazil. And I proceeded in going, "You're intelligent, na 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 na, you're intelligent." You get the idea? And she went crying to my mother. "Deanie called me intelligent." And my mother, "Oh, but that's a good thing." And she was, "No, it's not." So my mother, I was fairly amused and proud of myself for this, but my mother was less so. At this point, I should say that my sister is an incredibly well-adjusted and amazing human being, and that she's a compassionate therapist, and I admire her very much, and I don't think I did any permanent damage with those and other pilot experiments. But so from my teens, I knew I wanted to go into neuroscience in Brazil. Particularly at the time, I don't think we had any neuroscience majors, so I got my my BS in biology. And for my graduate work, I wanted to do experimental and computational neuroscience. So I was fortunate to be able to come to the U.S. and study with Jack Byrne at the University of Texas Medical School in Houston. And there I also worked with Michael Mauck. And with Michael Mauck, I published my first paper on timing, a model of timing in the cerebellum. And Mike Mauck was also a very valuable role model to me, somebody who I admired very much at the time. And then I went on to do my postdoctoral work with Michael Merznick. And I also published some papers on timing there, and a computational paper on what came to be known later as reservoir computing. I was fortunate to get my first assistant professor job at UCLA. And while at UCLA, my first years there, I would get a lot of papers to review on hippocampus and anterograde amnesia, which wasn't really my wheelhouse, and I was puzzled by this until I realized that my website said my research focus was temporal processing, to which many people understood processing in the temporal lobe. So I'm very upset to this day that the temporal lobe is undeservedly called the temporal lobe, because it doesn't really have any obvious, well-known special application in timing. So why is the temporal lobe called temporal lobe? Well, obviously it's because it's the temporal bone and the tempes, but why are the tempes called time in Latin, tempus? It turns out that, as I will now demonstrate, it's because the temples are an indication of the passage of time. So apparently that's why we have the temporal lobe to blame for why it's called timing, the timing lobe. So I knew since my graduate student days that I felt that in the neuroscience of timing and temporal processing was an important field, and that it was an understudied field. So back then, there were no biologically plausible models of how neural circuits could tell time. And we didn't even know basic questions in terms of whether timing is something that improves with practice. And there's always this gap between timing and between spatial processing and temporal processing. We learned about place cells before time cells. We studied spatial attention before temporal attention. We knew how neurons became responsive to spatial patterns before temporal patterns, and so forth. So I think it's worth giving a bit of a historical perspective of why timing and temporal processing lags spatial understanding and spatial processing. And if we go back, there's always been an appreciation of the importance of time. And say what you will about phrenologists, at least the phrenologists were on top of it enough to attribute to put in there this part right here, somewhere in the frontal lobe, a time area, right below mirthfulness, whatever that is. And in this area here is the office of faculty is to mark the passage of time, duration, succession of events, etc. It's also to remember dates, keep correct time in music, dancing, and induces punctuality in the fulfillment of engagements. This is an extremely centralized version of time, but surprisingly it persisted much longer than it should have in many circles. And I think it's too much of an exaggeration to say that there's some corners in which they still have this very centralized view that there's some part of the brain that tells time. And then, even William James in his magnum opus, Principles of Psychology, he had a chapter, the second to last chapter, on the importance of time and time perception. But you fast forward 100 years, over 100 years, and then you look at the principles of neuroscience, and you go to the index and you look for time, which is the most common noun in the English language. And time is not there. Temporal is there, but that's just because of the damn temporal lobe again. And so my point is not this is an omission of this textbook. My point is this is an omission of the field of neuroscience. And I think this is because neuroscience is a young field. And young fields, as they mature, they slowly incorporate time. The example I use here is that if you think about what was probably the first field in modern science, I would argue that it's geometry. So why is geometry, or why was geometry the first field of modern science? I think it's because it has no time. It's timeless. So geometry is the study of physical relationships, spatial relationships that don't change in time. It's much more complicated when you bring in change and dynamics. So it took 2,000 years and the likes of Galileo and Leibniz and Newton and eventually Einstein to really fully bring in time and dynamics into physics and mathematics. And it remains the case that time sits at the center of many unresolved scientific problems, from consciousness, free will, AI, relativity, quantum gravity, and the nature of time, which I'm not going to talk about, but I'll just give you a flavor of what I mean by this. And I think this is an example that many people find surprising is even a problem. So in the philosophy of time, there's sort of these two views. One is called presentism, and this is really the view that all of us have intuitively, that only the present is real. The past was real, and we have memory of the past, and the future is not yet real. And the actions we take in the present shape the future. The other view, the opposing view, is called eternalism, or the block universe, in which the past, present, future are equally real. So time is sort of spatialized, and the present just seems special because that's the moment we happen to be in. So in this view, now is to time as here is to space. So it's just one moment to be in or one place to be in. So this debate is actually, as obvious as this may seem to most of us, this is actually a debate that creates tension between physics and neuroscience. So here's Roger Penrose. It seems to me that there are severe discrepancies, but we consciously feel concerning the flow of time and what our theories assert about the reality of the physical world. This is Carlo Rovelli, the physicist Carlo Rovelli. I suspect that what we call the flowing of time has to be understood by studying the structure of our brain rather than by studying physics. So many physicists and philosophers have the default view that this is correct. And there's this tension arguing that, well, the neuroscientists have to figure out why we have this feeling of the flow of time if the laws of nature are telling us it doesn't flow. This is just an interpretation, I should say. This is a wide open debate. And Carlo and I sort of discuss this and the differences between the neuroscience of time and the physics of time in a chapter over here. And we conclude that really the only way to distinguish between these two theories anyways is if time travel were possible. So if time travel, somebody were to come back from the future, for example, and prove to us time travel is possible, that means we would live in an eternalist universe. Because under presentism, time travel is absolutely impossible because you can't travel to moments that don't exist. So all the science fiction books and movies we watch are implicitly eternalist in their view, because that's the only way time travel could exist. So I'm trying to raise funds to test this now. I can't emphasize enough how irrelevant this is to day-to-day science. So obviously, what my lab really studies is the neuroscience of time. So we look at the brain as an inherently temporal organ, and it's an organ that remembers the past to predict the future. Predicting the future and interacting with the external world requires the ability to tell time, decode temporal patterns, and generate temporal patterns. The brain generates the subjective feeling, this conscious feeling of the passage of time that's in some tension with physics, and the brain allows us to engage in mentally project ourselves into the past, to engage in what we call mental time travel. Now, my lab really focuses on point number 2 here. So how the brain tells time and generates temporal patterns, decodes temporal patterns, and so forth. And that's what I'm going to focus on in my talk. Because of the importance of time, the brain has mechanisms in place to tell time over 12 orders of magnitude. And unlike the clocks on our wrists, our watches, which are amazing devices that can tell time across, the single device can tell time in terms of microseconds to weeks, months, and beyond, the brain has fundamentally different mechanisms to tell time across these scales. So the circadian clock has nothing to do with our ability to tell time in music or speech. So they're fundamentally different mechanisms, and that's really important to understand. So what I'm going to focus on is in this time scale of tens of milliseconds to a few seconds, and that's the time scale we're interested in. Now, even in that range, I think it's important to distinguish between different types of timing. And I'm going to make the distinction between sensory timing and motor timing. So in sensory timing, you're discriminating temporal patterns that are coming from the external world. You're making decisions based on the temporal structure of a note or of music or of Morse code. And in motor timing, you're generating those temporal patterns, such as you would need to double-click a mouse, expect a change in the red light, or generate a musical pattern or a Morse code. And I think maybe this is, we could also think of this as sort of passive timing and active timing. And here under active timing, I would certainly include things like timing and attention, where you're actively creating a temporal expectation as to when something will happen. Now, as I said, when we started off, even the basic of the answer to a simple question such as, does interval discrimination undergo learning, was not known. So early studies we performed asked, in an interval discrimination task, does practice make perfect? So in a standard interval discrimination task, you would be given two intervals, each interval demarcated by a tone, so it sounds like this. So that first interval was 100 milliseconds, and then you'd be asked which of those were longer. In that case, the first one was the 100, and the second one was 150, so you'd say the second. And by changing that in an adaptive procedure, you could calculate people's thresholds, and the threshold might start off at 123 or 24 milliseconds, and then with practice, fall to 10. So this was one of the first demonstrations that whatever the mechanisms underlying learning, they improve with practice. What we didn't expect is that they'd be interval specific. Oops. So here, it turns out that if you practice at 100 milliseconds, you don't get better at 100, 200, or 500. So this was showing that you don't generalize across different intervals. Now, that was sensory timing. Now, in the motor timing, you also undergo learning. So whatever the neural mechanisms underlying motor timing are, they also improve. So in these tasks, you'd be given, this is a temporal production task. Where's my mouse? And you'd hear something like that. And then you'd tap that out on your keyboard. And before learning, we can just fit those to Gaussian curves here. And those curves, the standard deviation of those curves get tighter. We can plot those as standard deviation versus time and look at the slope. This is called the Weber fraction here, or the Weber coefficient is the slope here. And that improves with practice. But what, of course, we're really interested in how the brain solves these problems. How does the brain solve and perform temporal computations? And we've proposed for many years now that the brain relies on its inherent neuronal and neural dynamics to tell time, as opposed to, say, oscillators or pacemaker equilibrium mechanisms. That's number one. Number two, that sensory and motor timing rely on different types of regimes and neural dynamics. So I'm going to try to make the point that we should think of sensory and motor timing as potentially relying on two different mechanisms. So to understand this, it's first useful to state that we can think of the states of neural networks evolving in time based on two different types of measures. So the first, I'm going to refer to the active state. And that's what everybody's used to. So if we look at a neural network and we look at what neurons are active, here we have a spatial-temporal pattern, and we can look at a vector at a given time bin here, and that vector, a population vector, can change in time. So that's what we normally talk about when we're talking about the state of a neural network, the trajectory it forms in time. But in addition to this active state, there's what we can call the hidden state of a neural network. And that refers to synaptic and neuronal properties that are changing in time, but that aren't expressed, that you wouldn't pick up with an extracellular or intracellular recording. So if you look at a neural network and or neural circuit, in which even if the neurons are not doing anything, the state of the system can still be changing in time. So if it was recently active, it can still be changing in time due to, say, something like short-term synaptic plasticity. So here's an example of short-term synaptic plasticity. Pulses every, I think it's 40 milliseconds here. And the first pulse may be half a millivolt, then the second one is larger, then it starts leveling off. You pause for 500 milliseconds, and it's still very high. So it has a memory of what happened in the past. And so even when the network is off, there's information about the past, or temporal information, changing in the hidden state of this neural network. So what the contribution of these two states, the active and hidden state, to the information processing of a neural network is determined in large part by whether the recurrent neural network finds itself in what we call a low-gain regime or a high-gain regime. So what do we mean by that? All we refer to is, if you imagine this simplified recurrent neural network over here, and it has randomly connected weights, and those weights are shown here. They're around 0, so it's a balanced EI network, and the standard deviation is relatively small. That means the weights are pretty weak. So if we give it an input, nothing much happens. It's not much of a recurrent neural network because it's in the low-gain regime, and the activity doesn't propagate. So if we run that simulation, you give it the input, nothing happens. Now, if we're going to increase the weights a bit, so the standard deviation now becomes a bit stronger, now you get a bit of reverberation and perpetuating. Now we make them even stronger. Now you've totally shifted into a high-gain regime in which the input you give now propagates in perpetuity across the network forever and ever and ever and ever. This happens to be what we'll call, this happens to be a chaotic regime here, so this will just go on. So here now you can see that this network, certainly its active state, has information about the past, potentially. So our proposal, what we've sort of proposed over the years, is that sensory timing can rely on the hidden state of low-gain neural networks. So what does this mean? So let's go back to that interval discrimination task I just gave you, in which you have two tones, the first tone and the second tone. The first tone comes in, say we're looking at the auditory cortex, and it stimulates a bunch of neurons here, shown in white. Then there's a silent period, and no neurons are active in this hypothetical example. But because the hidden state is still changing in time, that means when the same input arrives 100 milliseconds later, it will arrive in a different state. So the same input should cause a different response here and here, and that allows you to encode information about time and previous information. So the idea here is cortical networks are inherently encoding not only the current stimulus, but the current stimulus in the context of the previous stimulus. And we've shown with models that you can account for these interval selective neurons that you observe in sensory cortex. So in sensory cortex, you can see in the auditory cortex, for example, some neurons respond better to specific intervals. So here's an interval of an interval-tuned neuron to two auditory tones. This is a duration-tuned neuron in the visual cortex, which it responds better to a bar of light that's 400 milliseconds. And we've shown with modeling work over the years that if you simulate a recurrent neural network in the low-gain regime that has short-term plasticity and give it a short interval or a long interval, you can generate interval-selective cells or cells that are tuned to specific intervals. This is what's now sort of called a reservoir network. Now, hopefully you can see that these networks cannot do motor timing, because in a motor timing, you have to actively generate a response at a specific point in time. And these networks in the low gain regime, after the input is off, won't generate something in the future. So there you need a high gain neural network. So here I'm going to give you an example of another trained recurrent neural network, again in a simplified implementation with firing rate neurons. And here are all the neurons, and we've sort of trained the weights here within the recurrent network and onto the output neurons, X and Y. And X and Y are just the motor neurons sort of positioned in two-dimensional space here. And the network starts running. All the neurons are sort of oscillating around zero activity. And then we give it an input, and that input will trigger a neural trajectory that will drive the XY neurons. And then that neural trajectory ends, and it just comes back to its baseline state. So this is what we call a population clock or a neural population clock. And the key part here is that it's an instantiation of what we'll also call a dynamic attractor. So these are networks in which generate these neural trajectories, but the neural trajectories are stable in that if you perturb them, they come back to the trajectory in motion. So in this example I'll show you here, we can have a recurrent neural network with random weights driving these two output neurons, X and Y, that write the word chaos in two-dimensional space here. And in one case, in both cases, we'll have trained these weights to produce the word chaos. But in one case, these weights will have been random, and the other, they'll be trained to implement a dynamic attractor. So here's that example. So in the top case, we inject a tiny bit of noise, and these are overlaid trials. And because this isn't a dynamic attractor, a tiny bit of noise will diverge exponentially, and you'll lose track of what you were doing in the past. You'll lose information about what happened in the past. In this case, we're injecting a lot more noise. So you can see this perturbation is way larger than that perturbation, but the network finds its way back to its original trajectory. So what we're saying is we have a trajectory in high-dimensional space. You kick it off that trajectory, and it sort of converges back to this sort of tube of attraction rather than a basin of attraction that we talk about in standard dynamical systems. So in vivo recordings during timing tasks, people have observed many flavors or many regimes of what we think are neural trajectories in these dynamic attractor regimes. And that can be these beautiful, very sparse neural sequences in the birdsong, or less sparse sequences in the striatum or hippocampus, ramping activity in the striatum, or just complex activity in different cortical areas and even in human hippocampus. Now, what was striking to us is that all these regimes that have been implicated in timing over the years have also been implicated in working memory. Now, the standard view of working memory is that, going back to Fuster and Patricia Goldman-Rakish, is that maybe working memory is stored in the language of dynamical systems, a fixed point attractor, right? That neurons go on, they stay at some sort of fixed steady state activity, and then they'll go off when you use that memory. But over the decades, people have observed many other different regimes of associated with working memory in different brain areas, including ramping neural sequences, complex activity, and even what some groups call activity-silent networks or mechanisms for working memory that rely on short-term synaptic plasticity. So there's been almost this perfect convergence between the models of working memory and timing. So we were interested in asking, well, is this a coincidence? Or maybe working memory and timing are, in some cases, mirror computations of one another. So what do we mean by that? So timing and working memory share certain features. They both transiently require storage of information. So if we think of a simple hourglass, and we see its current state, that has information about the past, that somebody turned that hourglass over, or an egg is being boiled, but it also has prospective information in terms of when something should happen in the future. So this has a component of both working memory and timing. So we were interested in studying this, the relationship between working memory and timing. And these studies are in many ways inspired from some of Kia's work, who's done some beautiful work on implicit timing and the relationship with working memory. So in this task, what we're going to call this differential delay match to sample task is your standard delayed match to sample, in which you have A, B, B, A, B, B, and match, non-match, non-match, match. The only difference is that A will cue a short delay, and B will cue a long delay. These is task irrelevant, right? Because the delay is, strictly speaking, irrelevant to the task that's being performed. So we want to know, is there an interaction between the working memory component and the implicit timing component? And to do that, again, as Kia's done in many of her studies, then you just do a reversal condition in which now you break the expected relationships. In humans, we found that performance dropped in the unexpected conditions in which A predicted A, which was associated with a short delay, is now generating a long delay, and vice versa. So here's this inverse efficiency, which is reaction time over accuracy. So that's the human data. And we've wanted to know with animal data to record during these tasks. So we did this in collaboration with Payman Golshani's lab and a graduate student, Connor Dorian. So in the mouse version of this task, you have two odors, A and B. And again, A predicts a short delay and B predicts a long delay. And then you have the standard conditioning, which all the training is done on, and then you have a testing situation. And again, the animals implicitly learn the timing. That is, when you change the delay, the task irrelevant delay again, performance drops in terms of percent correct. It's a slight drop, but significant one. But in the case of the mice, all of that drop is driven by cases in which they expected a short delay but received a long delay. In other words, if they were expecting long and received short, their performance was normal. But if they were expecting short and received long, then performance dropped. So this makes one wonder is if working memory has an expiration date. So if when we store items in working memory, we also learn to store that information for a specific point in time in a task-dependent manner. Now, these are still early days on the recordings, but initially what we've done is recorded in CA3. So this is calcium imaging in CA3 during the task. And as other people have observed, is we can see the onset responses to the odor. These are the odor selective neurons, and then you see these delay neurons that are firing during the delay, and here they're sorted according to when they fire. These neurons are sorted during the short delay. These are neurons during the long delay, and there's very little overlap between the neurons here and here. So these are totally different neural trajectories. So the question is what happens now when you change the delay? So here it's obvious, right? So now you're going to change the delay. It's expecting long. You do it short, so you just have to cut this short. But what happens over here? So now you're going to make this longer than expected, and you normally have the neural sequence, but now it's longer than expected, and the sequence sort of runs out. It sort of gets reaches its final destination in trajectory space and sort of runs out. So we know that the structure of these neural trajectories and these neural trajectories are different, which establishes that they're not simply encoding working memory. Whatever they're encoding is working memory in a manner that's dependent on the temporal structure of the task. To further make that point, if we try to decode time now by just decoding time in the short interval, in the short window here in both the long and the short delay, decoding of time is improved, is better here compared to here. So there's a structure in the differential structure of the neural dynamics of these two trajectories. So we take this as suggesting that in some cases, timing and working memory may be multiplexed, as it might be advantageous for some neural representations to encode working memory and when that information will be used or for how long it will be used. So we're raising the possibility that working memory and timing might be encoded in the same neural representations. Okay, now everything I've said up to now is sort of implicit in the idea that timing is a distributed phenomenon, that neural circuits can tell time on an as-needed basis. Basically what we're suggesting is timing is so important that it wouldn't make sense to have a centralized area devoted to timing, but that most neural circuits, particularly neocortical circuits, can tell time on an as-needed basis. In other words, what we're suggesting is that timing is a universal computation of neocortical circuits and that it's a computational primitive. So how can you address this? So one approach we have taken is to ask, well, can we teach cortical circuits ex vivo in a dish to learn temporal patterns? Because if it is a computational primitive, then that should be possible. So how are we going to go about doing this? We're going to use organotypic cultures, and we're going to transduce neurons in those cultures sparsely with one of two options, the channel rhodopsin or crimson. So we'll have some blue neurons and some red neurons. This is just showing that we can have two input channels. So these are two simultaneously recorded neurons, a crimson neuron and a channel rhodopsin neuron, in response to red light or blue light. So you can see when we give the red light that you get these robust responses in the red cell. When you give the blue light, you get robust responses in the blue cell. But there is some crosstalk here, by the way, and that's well known, and we knew that was going to happen because crimson has a long left tail. And so although its peak is around 600 or 590 nanometers, it can be activated by 450 nanometers. But the bottom line is there's a differential activation of the red and the blue neurons. So now we asked, well, can we train these with different temporal relationships? So we're using basically a very simple task, which is sort of like a delay conditioning task. So we'll call the red light a CS, a conditioned stimulus, and we'll call the blue light a US, the unconditioned stimulus. And all we're going to do is change the interstimulus interval. So we're going to have one group in which you have a short interval and one group in which you have a long interval, both delay conditioning. And then during the test conditioning, you just give the CS again. And we want to record from neurons in that slice and determine if there's differential responses reflective of learning. So this is work by Ben Liu in my lab. And this is an example of a cell trained. So he's now recording from opsin negative neurons. Okay, so he's recording from opsin negative neurons, shown here, and giving it red light, and seeing that in the early group, this neuron responded relatively early, has an onset response, and then it spikes early. Whereas this neuron from the late training group had an onset response and responded late. This is the average data. So these are all the early cells, and these are all the late cells, and there's this very, very distinct temporal profile to them, as if the late cells were anticipating the arrival of the blue light, or predicting, if you will, the arrival of the blue light. So this establishes that the circuits in these ex vivo preparations are certainly being shaped by the stimuli they're being exposed to, and in a sense, learning. This is just the average data, or all the raw data across all slices and trials from the early and late group, and just the differential timing here. So there's very dramatic difference. Now, what we didn't expect is that these same slices have spontaneous activity. That was well known. What we didn't expect is the spontaneous activity is different in the slices as well. And it turns out that they seem to be replaying spontaneously the temporal structure of the stimuli they were exposed to. So this is not evoked activity. There's no red light here. Now we're just recording from opsin negative neurons, hanging out, waiting, and see what the network does. And on average, early cells in the early trained group have these very rapid onset responses, and that very short lasting. But in the late group, they again generate responses that look a lot like the evoked responses. So to our surprise, the tissue seems to be replaying the evoked activity. So this is the example of replay that's been observed in vivo, of course. So this is the average data across all the early cells and across all the late cells, and there was a significant difference in the time to peak. Now, we can also, to further explore this replay, start looking at the mechanisms. And if you get an untrained slice and record from a crimson neuron and a channel rhodopsin neuron, and look at the spontaneous activity, we also call these upstates, they're highly synchronized. This is what we expected, because there's no reason it wouldn't be, because during an upstate, all the neurons go on together. So there's a very tight time locking between the blue and the red cells. But in the late group, the late trained group, the red cells generally preceded activity in the blue cells. Remember, this is spontaneous activity now. So the only reason that this could be happening is because it was dependent on the experience of that slice. And so there was, again, a significant difference in the correlation. That's just shown here. The correlation was much higher in the untrained red versus blue neurons versus the trained red versus blue neurons. Now, an interpretation of these late responses is that they could be anticipating the blue light, or also another interpretation is they could be coding for a prediction error. So what do we mean by that? You can look at this as saying, well, it's sort of anticipating the blue light, but you notice that most of this activity is happening late. The blue light would be happening right here. So maybe a better interpretation here is that this is a prediction error signaling the absence of the expected blue light. So to address that, in a subset of neurons, we compared the response to the red light plus the blue light. And some subset of neurons responded less to the red plus the blue light. Basically, the blue light was inhibiting the response. So these yellow neurons here, 25% of the neurons, might be consistent with what you would call a temporal prediction error, or just a prediction error. Now, everything I'm saying up to now is really contingent on the notion that the circuit is learning to tell time, that the circuit is tuning its properties to generate these late responses. Now, what I'd like to explore is a more fundamental question pertaining to the synapses themselves. So can synapses learn to tell time? That is, can they tune their synaptic dynamics to optimize the processing of temporal information? So traditionally, we look at synapses as computational elements that are tuned with one scalar variable, one scalar parameter, to be weak or strong. It could be 0, very weak, or very strong through long-term plasticity. And whether it's a complex artificial neural network or the neocortex, we look at the information processing abilities of these networks to rely on the tuning of these W's here, the WIJs. And that these WIJs, as I said, have one parameter, the synaptic weight. What we're suggesting is that synapses are more complex computational elements than this standard tenet in neuroscience. We're suggesting that synapses don't simply learn to be weak or strong, but learn when to be weak or strong. So they have more than one tunable parameter. So of course, this brings us back to short-term synaptic plasticity. So here's two examples of short-term synaptic plasticity. Here's data from the Allen Institute. These are layer 2-3 pyramidal neuron recordings, paired recordings from the Allen Institute data. This is data from humans, by the way. And this is an example of short-term depression. The first EPSP goes up. The second ones get smaller and smaller and smaller. Then there's a recovery period, and it sort of recovers. Here's an example of short-term facilitation. This is the same example you saw before. Now, short-term plasticity, short-term synaptic plasticity, is generally modeled. The most common is modeled in different ways, but the most common model is what we call the Tsai-Markram model. And in the Tsai-Markram model, the idea is that short-term synaptic plasticity is a presynaptic phenomenon, and it basically relies on this balance or this push-pull mechanism between vesicle depletion and vesicle replenishment and probability of release. So in the standard model, Tsai-Markram model, there's this D for depression variable, and that just refers to the depletion from the readily releasable pool, and that recovers with a time constant tau D. Facilitation, which could be interpreted as residual calcium in the presynaptic terminal, can go up when there's a presynaptic spike. So F can go up, and that will increase the amount of transmitter release, and that will recover or decay with tau F. So the Tsai-Markram model has three parameters, tau D, tau F, and U. And U is sort of the amount of transmitter release, a bit related to probability of release. So what we're proposing now is that in addition to the standard W in the synapse, that these three parameters might be able to be learned as well. So this is just showing computationally that short-term synaptic plasticity now increases the computational power of synapses. So now a single synapse can do an interval discrimination task. So if we now have a neuron that we want to respond to a short interval, but not to the medium or the long interval, we can tune, and here we're just using tuning with stochastic gradient descent, so non-biologically plausible, and we can tune these parameters so this neuron will fire here. Or we can tune it so that this neuron will fire in the medium interval or at the late interval. So you arrive at different combinations of U, tau D, tau F. Similarly, you can now, by using this learned synaptic dynamics, you can now have each synapse essentially implement a counter. So now if the presynaptic neuron is firing at 5 spikes, and now our postsynaptic neuron can fire at the early, second, third, fourth, or fifth, depending again on these three presynaptic parameters governing STP. So we can train it to be an interval counter, if you will. So this is just showing that in principle, extending synaptic plasticity into the temporal domain by making short-term synaptic plasticity plastic, what we're going to call learned synaptic dynamics, enhances the computational power of synapses and neural networks. So we're proposing that this is learned. So let me just give you a quick reminder of what we mean by learned and what we mean by a learning rule. So what we mean by a learning rule, the most famous being Hebb. Hebb told us that if you have a presynaptic neuron and a postsynaptic neuron, and the presynaptic and postsynaptic neuron fire together, neurons that fire together, wire together, the synapse gets stronger. STDP, spike timing dependent plasticity, extended that by showing the precise temporal relationship, expanded on Hebb, by showing that the precise temporal relationship between the pre and postsynaptic neuron matters, saying that if the presynaptic neuron fires before the postsynaptic neuron, then you get potentiation. But if the postsynaptic neuron fires before the presynaptic neuron, you get depression. So these are two of the most common synaptic learning rules, and their associative forms of synaptic plasticity. Now, neither of these models mentions short-term synaptic plasticity. So what's happening to synaptic dynamics on the time scale of tens to hundreds of milliseconds? What we're proposing under the learned synaptic dynamics hypothesis is that if you have a presynaptic train, a presynaptic sequence of action potentials, and the postsynaptic neuron fires late, that synapse wants to learn to have facilitation. If it fires early, that synapse wants to learn to have depression. So hopefully you can see the logic here, right? The synapse, you're extending Hebb, because this is causally involved with the firing here. Here, the causality is early. Here, the causality is late. So what you want is that the synapse have maximum potency at the time of the postsynaptic spike. So here, if it was sort of a computational efficient system, you'd want this synapse to learn to have facilitation because it's not firing early, and here to have depression because it's not firing late. Okay. So now we're going to test this hypothesis. Again, this is very new data. And we're testing the learned synaptic dynamics hypothesis. And we're using basically the same protocol you just saw. But rather than look at the circuit properties, we're looking at the EPSPs being evoked by the red cells onto the channel rhodopsin cells. So we do the same training protocol. And during testing now, we've partially blocked recurrent activity, or to dampen activity again, or else the recurrent activity over masks the EPSPs that are hard to see. So this is an example. These are one of our best examples, of course, in which we're showing in a late trained case that the cell exhibited facilitation in response to red light, and here depression. This is the average data. So on average, the first EPSP amplitude was the same between the early and the late, but while there was depression, the temporal profile here and temporal profile here are different. These are a bit misleading because you have to actually calculate the EPSP amplitude by subtracting the temporal summation. So it's better to look here. So here you have dramatic depression in the early group, and here in some cells had facilitation, some had mild depression, but on average they were pretty flat. So the point is that the temporal profile of STP was different according to the training experience that those two slices underwent. Now, to test if that's synapse specific, we can do the following experiments. Again, this is the training case in which now we're just stimulating red light and recording in the blue cells. And that's the same data you just saw. And here, it's the same slices, but we're just recording in the opsin negative cells. So if it's synapse specific, we should see different temporal profiles when we're looking at this synapse versus that synapse. And that's what we see. So in the opsin negative cells, we see no difference between the temporal profile of short-term synaptic plasticity, and here we do. So these are just our first indication that synaptic plasticity itself, or short-term synaptic plasticity, might be learned, and that synapses are computational elements that we've really missed an important component of the computational power of synapses. Okay, so let me wrap up here and summarize. So because of its importance, we propose that timing is a highly distributed computation that relies on intrinsic dynamics of neural circuits. Sensory timing on the low recurrent network regimes, low-gain recurrent neural network regimes, and short-term synaptic plasticity, and motor timing on stable neural trajectories, dynamic attractors, which would be the high-gain regimes. In support of this notion that timing is a highly distributed computation, a computational primitive, if you will, is that even cortical circuits in a dish can learn simple forms of timing and temporal prediction. And lastly, we now have preliminary evidence that synapses may be more complex computational elements than previously thought, that they might not only learn to be weak or strong, but they might learn to tune their temporal profile of synaptic dynamics to peak at the time of the postsynaptic activity. Lastly, let me just thank my lab. I mostly talked about Ben Liu's and Jamie McDowell's research here, but Tsai contributed as well. So thank you.